All right, welcome to Chem 313, Lecture 8. To review from Lecture 7, very briefly, we looked at how it is that enzymes can lower the activation energy for a reaction. And we said that there are certain things that enzymes generally do when they are catalyzing a chemical reaction. We said that enzymes bind to the substrates. They orient the substrates so that they are likely to react. And then on top of that, they directly contribute to lowering the activation energy by one of several possible features. Probably the most common way is by binding to the transition state of the reaction, and that is by forming non-covalent intermolecular forces with the transition state. In some cases, the enzymes provide an alternate mechanism for the reaction. And the idea behind providing an alternate mechanism is that maybe there's another possible mechanism that has a lower activation energy than the one that would be present without the enzyme. And so it can do this by acting as an acid or a base. And we saw that in malate dehydrogenase in lecture 7. We saw that there's a histidine that's acting as a base. It may form covalent bonds with the substrates. We did not see an example of that, but the textbook does talk about that somewhat. And it may also donate or accept functional groups, for example, an amine or a carboxylic acid or a methyl, whatever. Or, I didn't mention this last time, it may also accept or donate electrons to a substrate. So we can imagine a situation where in the absence of an enzyme, our substrate would have this much free energy. We go over this energy barrier and our product would have this much free energy. Using an enzyme is more complicated, but typically what happens is we replace the big internal hill with a couple of smaller hills. We have one small hill which is related to enzyme substrate binding, so we form our enzyme substrate complex. And then we have our new activation energy for the reaction, which is lower in free energy. And finally, we end up with our product, which has the same free energy that it would have in the absence of any reaction. So this is for the reaction scheme, enzyme plus substrate react to give us enzyme substrate complex, which then immediately falls apart to give us enzyme plus product. So in the absence of an enzyme, we would have this much activation energy. In the presence of the enzyme, we have only that much. Today's lecture deals with enzyme inhibitors. So enzyme inhibitors may seem like an artificial subject. Why would we care about something that inhibits enzymes after I've just spent all this time talking about why enzymes are great? Well, enzyme inhibitors have a lot of important properties and characteristics. These are important for a lot of drugs. So many of the drugs out there are enzyme inhibitors. Many poisons or naturally occurring toxins are poisonous because they inhibit enzymes. They've got all kinds of industrial relevance as well. And then for a less obvious application of enzyme inhibitors can sort of act as a way for living cells to control the activity of enzymes so that reactions only happen when and where they're needed. And we probably will see some examples of that later in the course. Now, what I expect you guys to take from this lecture, I don't care that you know in exquisite detail what each and every inhibitor does. I think you should be able to know the differences between the inhibitors in terms of how they work and their effects on the Km or Vmax of the enzyme. Why they have that effect. And then lastly, I'm fond of asking questions about what is some sort of an experiment that you could do that could tell you what type of inhibitor a molecule is. And if you understand the first three, I think that last one will be much easier than it sounds. So let's talk about enzyme inhibitors. I like to go into some depth on enzyme inhibitors because I think they help reinforce our understanding of how enzymes work. I think that's actually more important than the inhibitors themselves. So what are inhibitors of enzymes? So these are non-enzyme molecules okay, usually but not always small organic molecules that interfere with enzyme catalysis somehow. That's a very broad definition. And most inhibitors, not all, are specific to one or a few enzymes. So just as we said before that there are no general purpose enzymes, there's no enzyme that can catalyze any sort of chemical reaction you can name, there's no inhibitor that can inhibit any enzyme that you name. We have two broad classes of inhibitors, and we're going to spend most of our time talking about one class. We have irreversible inhibitors, and we have reversible inhibitors. And the names are pretty much self-explanatory. Irreversible inhibitors are permanent. Once these come into contact with your enzyme, it's dead forever until it's chopped up and recycled for amino acids. Reversible inhibitors are not permanent. They go away when the inhibitor goes away. And then there's several subclasses of reversible inhibitors. We've got competitive inhibitors. We're going to talk about each one of these separately. We've got so-called non-competitive inhibitors. We've got mixed inhibitors, which is a combination of those two in some respects. And then we've got a weird class called uncompetitive inhibitors. And we'll talk about that one later. And then lastly, we have allosteric inhibitors, and we've already pretty much seen those when we were talking about cooperative binding proteins. So the key difference between irreversible and reversible is the following. If the inhibition of the enzyme goes away, when the inhibitor is removed, you have a reversible inhibitor. If it does not, you have an irreversible inhibitor. Now, why does inhibition go away when the enzyme inhibitor is removed if it's reversible? Well, the reason it goes away is because regardless of what type of reversible inhibitor that you have, all of our reversible inhibitors bind to enzymes by weak non-covalent intermolecular forces. And because these weak non-covalent intermolecular forces are weak, binding is reversible. Irreversible inhibitors, for the most part, there are some exceptions, but they usually react chemically with the enzyme. And since covalent bonds are essentially permanent, we say that irreversible inhibitors act permanently. We're going to talk about irreversible inhibitors first. So for the purposes of this class, yes, irreversible inhibitors are permanent. And usually, not always, but usually what happens is the inhibitor reacts chemically with the enzyme to form an inactive product. So I show that conceptually with these little block diagrams. An inhibitor molecule will react with an enzyme to give us a non-functional enzyme inhibitor adduct. So what I've shown here is the case where the inhibitor reacts at the active site and then it's permanently stuck there. And if it's permanently stuck there, that means that the substrate can't bind. But that's just an example. So not all irreversible inhibitors work this way. Sometimes the reaction is somewhere else besides the active site. So an alternate possibility is that it modifies chemically some amino acid that is necessary for catalysis. So for example, maybe the active site of your enzyme has a cysteine, and the cysteine is necessary for catalyzing the reaction. And if your inhibitor reacts with that cysteine to, I don't know, put a methyl group on it, then your enzyme is going to be non-functional. Either way, what you end up with is a non-functional product. By non-functional, I mean it either can't bind the substrate anymore, or can't catalyze the reaction anymore. And this is a permanent thing. So what is the effect of an irreversible inhibitor if you mix it with a solution of enzymes? So let's say that you have a test tube full of enzymes and you add some irreversible inhibitor to it, and what's going to happen is you'll get a reaction and some fraction of your enzyme is going to be converted to this non-functional product. Well, what that means is that in effect, it's as if the concentration of your enzyme goes down. Not literally, but if there's a big fraction of your enzyme that is non-functional, then for all practical purposes, it's as if that enzyme isn't even there, right? So in effect, the concentration of your enzyme in solution is going to decrease. Make sense? Hopefully it does. Now, if the concentration of enzyme in effect decreases, what effect do you think that's going to have on the Vmax of the enzyme? Well, we said before that Vmax is equal to kcat, the rate constant for catalysis, times enzyme concentration. So that means that an irreversible inhibitor causes Vmax to decrease. Now, what effect do you think this would have on the Km? Well, the answer may surprise you. Irreversible inhibitors, generally speaking, there are some rare exceptions, but for the most part, they have no effect on the Km of the enzyme at all. And that may seem weird because we said before that Km tells us about binding. And we said that what we've done, at least in some inhibitors, is we've modified our enzyme so that it can't bind the substrate anymore. Well, sure, but if your enzyme can't bind the substrate anymore, at that point, it's not really an enzyme anymore. Right? So if it's not really an enzyme anymore, it doesn't make sense to talk about the Km, right? Any enzyme that did not react can still bind substrate just fine. So for the most part, irreversible inhibitors will lower the Vmax for an enzyme, but they don't affect the Km. You just need more enzyme to get to a certain velocity. All right, this enzyme, cyclooxygenase. This enzyme is probably the best known example of an enzyme that is irreversibly inhibited. So cyclooxygenase is a very big, complicated molecule. It's involved in converting this substance called arachidonic acid, which is a fatty acid, we'll talk about those later, to prostaglandins. I think it's prostaglandin A, but I don't remember off the top of my head. And prostaglandins are important molecules in mediating inflammation. Okay, so cyclooxygenase is irreversibly inhibited. So what happens in this enzyme is there's this little tunnel that leads to the active site of the enzyme. It's hard to see in the structure here. But in this tunnel, there's a serine, right? And this serine can react with this drug, which is aspirin. And what ends up happening is the aspirin's acetyl group gets transferred onto the OH so that we're left with a molecule that looks like this. And we're left with regular old salicylic acid, which is the byproduct, and that can just be excreted. So what we end up with is this acetylserine, and this acetylserine sterically blocks the active site. Just because it's too big for the arachidonic acid substrate to get through. So this acetyl group blocks this tunnel and makes it so that cyclooxygenase is incapable of binding arachidonic acid. And it's a permanent reaction. So the enzyme is dead and it stays dead until it gets chopped up for scrap, so to speak. So because of this, we have a useless enzyme, we don't get our prostaglandins, and we don't get inflammation. Aspirin is just one of a class of drugs called nonsteroidal anti-inflammatory drugs. And so that includes things like naproxen, your over-the-counter pain relievers, most of them. And they all work the same way. They chemically modify cyclooxygenase. Okay, let's talk about reversible inhibitors now. So remember, reversible inhibitors, they simply bind to the protein non-covalently. And the difference between the different reversible inhibitors mostly boils down to where they bind and what happens when they're bound. So the easiest and most common and most conceptually simple to understand type of reversible inhibitor is the competitive inhibitor. So the definition of a competitive inhibitor is that it binds to the same spot as the substrate. And obviously, if it binds to the same site as the substrate, then that means that the substrate can't bind if the inhibitor is bound. So I tried to symbolize that by using color-coded spheres. So we've got our enzyme, our substrate is green, our inhibitor is purple. And when you have a free enzyme, either the inhibitor can bind or the substrate can bind. Now, once the substrate binds, the reaction can go normally. But if the inhibitor is bound, you have no reaction taking place. Now, both of these processes are reversible. So that means that even if an inhibitor is bound, it can still unbind and you can go back to binding your substrate and having a reaction. So in effect, what happens is that the inhibitor and the substrate compete for binding, which is where the term comes from. All right, so if the inhibitor and the substrate compete for binding, what does that mean? That means that the binding of the substrate is less effective. It's not impossible because binding of the inhibitor is reversible. But the more inhibitor that there is lying around, the less likely the substrate is going to bind because the inhibitor is more likely to be bound in its place. So if the substrate binding becomes less effective, what happens to our Km? Our Km increases. So you can see that in a Michaelis-Menten graph. If you have a competitive inhibitor, your Km is going to increase. This would be our Km. This would be our Km with the inhibition. But notice the way I drew this diagram. What do we see? We see that our V curve levels off at the same Vmax. And why is that? Well, the reason for that is if we end up in a situation where our substrate concentration is much greater than our inhibitor concentration, well, that means that you've got so much substrate that the enzyme is more likely to bind to the substrate than to the inhibitor. And because of that, Vmax is not affected by a competitive inhibitor. Another way of putting that is that the inhibition can be overcome or outcompeted at high substrate concentrations. You have the same Vmax. All right, so to summarize, Km goes up, Vmax does not change. This is what would happen to your Michaelis-Menten graph. A couple lectures ago, I mentioned that we teach the Lineweaver-Burke graphs because they're handy in certain applications, and enzyme inhibitors is one application. Lineweaver-Burke graphs make it obvious at a glance what type of inhibitor you have. This is what a competitive inhibitor does to the Lineweaver-Burke graph. Remember, the slope of a Lineweaver-Burke graph is equal to Km over Vmax, and the y-intercept is equal to 1 over Vmax. So you'll notice that on the Lineweaver-Burke graph, the y-intercept does not change because the Vmax does not change. But the slope increases because the Km has increased. So on a Lineweaver-Burke graph, the slope goes up, but not the y-intercept. So one last feature of competitive inhibitors that you've probably guessed by now, and I sort of hinted at with the way I drew the structures over here, is that if the competitive inhibitor and the substrate have to bind at the same place on the enzyme, what does that tell you about their structure? What that usually tells you is that they're structurally very similar. So competitive inhibitors often, not always, resemble the substrate. All right, I'll give you guys an example of a competitive inhibitor, probably the best known outside the world of medicine. Glyphosate, better known as Roundup, that's a competitive inhibitor. So plants have this enzyme called EPSP synthase that's used to make aromatic amino acids. So they form this complicated looking molecule called 5-enol pyruvyl shikimate 3-phosphate. Who cares? That goes on to form your aromatic amino acids. So humans do not have this enzyme. We have to get aromatic amino acids from our diet. But the key feature of this enzyme is that one of the substrates for the enzyme, phosphoenol pyruvate, which is shown over here, this is what the structure of phosphoenol pyruvate looks like. This is the structure of glyphosate. So you'll see that at a glance, they look very, very similar. They both have a phosphate on one side. All right, strictly speaking, it's a phosphonate on glyphosate, but who cares? They got a carboxylic acid on the other side. What's in the middle is a little bit different, but that doesn't matter. What's important is that these bind to the same site, but glyphosate can't react. And so what it does to plants is it starves them of aromatic amino acids, essentially, because it's inhibiting EPSP synthase. And because humans don't have this enzyme, I think most animals don't have this enzyme, this weed killer is not terribly toxic to animals. It's not exactly the most safe thing in the world, but it's not highly toxic the way other weed killers are. All right, a more medically relevant example is ritonavir. So it's a competitive inhibitor of HIV protease. So what it does is it functions as an antiviral. We'll put a couple letters there. So what does HIV protease do? So the HIV virus, when it infects a cell, it synthesizes this very long polypeptide. And essentially, this long polypeptide has to be chopped up into little segments. And those little segments fold up to form all of the virus's important proteins. So the protease cuts this big polypeptide. So the substrate of the enzyme is a protein. And if you look at this molecule closely, you'll notice that it kind of looks like a protein molecule. What do we got right here? Oh, we've got a peptide bond. And then we've got a branch side chain. That happens to be valine. We've got another peptide bond, right? We got another peptide bond down here. But then in between, things get a little bit screwy. So we don't have peptide bonds here or here. So what we have is something that's called a peptidomimetic. Which is a molecule that looks like a protein, but isn't. And what do we see? So we've got these valine and then a phenylalanine. I put in quotes because it's not technically a phenylalanine. There's no peptide bond there. And then another phenylalanine. So this binds to the active site of the enzyme, and the spot where the protease would normally cut is valine, phenylalanine, phenylalanine. And typically it cuts, I believe, right between the two phenylalanines. But since we don't have a peptide bond here, this can't be cut, and it just sits there in the active site of the enzyme and prevents the enzyme from ever cutting the HIV polypeptide. All right, next type of reversible inhibitor, non-competitive inhibition. So non-competitive inhibition is pretty unusual. It's something that we teach as sort of like an abstract hypothetical. What's more common than this is mixed inhibition, which I'll talk about in a second here. But anyway, the idea of a non-competitive inhibitor is that the inhibitor binds away from the active site and somehow prevents catalysis when it is bound. So if you want to have a cartoon representation, here it is. You've got your enzyme. It can bind to the substrate, or it can bind to the inhibitor. And when it's bound to the inhibitor, it can still bind substrate, but it can't react. It can only react if the inhibitor is not bound. So the inhibitor prevents catalysis when bound, but does not affect substrate binding. All right, so this situation is kind of the inverse of competitive inhibition. In competitive inhibition, we said that the inhibitor interferes with substrate binding but doesn't affect catalysis. Non-competitive inhibitors affect catalysis but not substrate binding. So what do you think would be the effect of these things on your Vmax? So substrate binding is not affected, catalysis is affected. The Vmax goes down. But we said substrate binding is not affected, so the Km is not affected at all. So again, that may make little sense because you may be thinking, well, wait a minute, if the substrate binds to the inhibitor enzyme complex, that binding isn't effective because it doesn't have any reaction taking place. Well, no, a better way of thinking of it is that if the inhibitor is bound, the enzyme is essentially dead. And so you can think of it as a temporary reduction in your enzyme concentration. So this is what we see in our Michaelis-Menten graph. We get a lowering of our Vmax, right? These curves level off at the same, or excuse me, at different Vmaxes, but the shape of the curve is the same. So if you were to trace over to this curve to find its 1/2 Vmax and do the same thing to this one, they would overlay. but we have different Vmaxes. So our Vmax decreases. Km has no change. So in that respect, it's just like an irreversible inhibitor. Irreversible inhibitors do the exact same thing to the Michaelis-Menten graph. So I said at the beginning of class, you know, what's an experiment that you could do to tell what type of inhibitor you have? Well, think about it. I'll give you guys one hint, and then if you wanted to check your work with me at office hours, please do that. You're reminded that non-competitive inhibitors are reversible. Now, let's take a look at the Lineweaver-Burke graph. What do you think that a non-competitive inhibitor would do to a Lineweaver-Burke graph? So what it does is it changes both the slope and the y-intercept. They both go up, right? And that's because, remember, our Km is changing, excuse me, our Km is not changing, but our Vmax is decreasing. And since our slope is Km over Vmax, and the Vmax is what changes, then both the slope and the y-intercept are going to change. So this is an enzyme called thymidylate synthase, and it's involved in synthesizing this molecule DTMP. So this is not the substrate, this is the product. The substrate looks exactly the same as the product does, except it doesn't have this methyl here. So the substrate and product basically look the same. So anyway, cells synthesize a lot of DTMP when they're synthesizing a lot of DNA, and this tends to happen a lot in cancer, obviously, because the cells are constantly dividing. So thymidylate synthase is subject to this molecule called raltetrexate, which is a non-competitive inhibitor that is used as a chemotherapy drug. Now you'll notice that the substrate and the inhibitor are not structurally similar at all. And that's because this inhibitor binds to a different spot on the molecule. I don't have a dedicated slide for it, so I'm just going to write in the margin over here. There's another type of reversible inhibitor called a mixed inhibitor, and that combines features of both competitive and non-competitive. So in mixed inhibitors, the inhibitor binds away from the active site, and when it's bound, both substrate binding and catalysis are inhibited. And usually that's because it induces some sort of a conformational change in the enzyme, so that it no longer is in the correct shape to bind to the substrate. And so what do mixed inhibitors do kinetically? Well, because you're interfering with catalysis, Vmax is going to go down, but you're also interfering with substrate binding, so Km goes up. Mixed inhibitors are actually more common than pure non-competitive inhibitors, but I don't have a dedicated slide for that. You can just think of a mixed inhibitor as a combo of the two previous. All right, the last type of reversible inhibitor is the weird one, uncompetitive inhibitors. This is the one that you just kind of have to know, because wrapping your head around why it works just isn't easy. The textbook tries valiantly to explain it, but I don't think it does a very good job. So the key feature of an uncompetitive inhibitor is that the inhibitor only binds to the enzyme substrate complex. So what happens is that the substrate binding is normal, but a fraction of the enzyme substrate complex is not going to be able to react. So if you want to think of it graphically, the enzyme and the substrate bind normally, but then after they bind, here comes the inhibitor. It binds the substrate complex and it locks it up so that it can't react. The only way it can react is for the inhibitor to come off, which it can, and then you have just a regular enzyme substrate complex again. So the effects on the kinetics of this one are, again, this is not easy to wrap your head around, so I'm just going to tell you what it is and just know it and you'll be fine. The Vmax goes down and the Km also goes down, but they go down by the same factor. So what that does to your Michaelis-Menten graph is it does something like this. It lowers the Vmax, but it changes the shape of the graph as well. Still hyperbolic, but now it's a shallower rise. So you may be thinking, well, wait a minute, the Km going down, how is that an inhibitor? That means that it's making the substrate binding better. Well, actually, no, it's not making it better. It's an illusion, because notice how I drew the green curve. The green curve is below the red at all substrate concentrations. So yes, it takes less substrate concentration to get to your new Vmax, which is lower, but because that new Vmax is so much lower, the curve in the presence of an inhibitor is below the curve in the absence of an inhibitor, no matter what concentration of substrate that you have. So it still counts as an inhibitor. So that's what it does to the Michaelis-Menten graph. What it does to the Lineweaver-Burke graph is very characteristic. It does not change the shape, the slope of the line, but it causes your y-intercept to increase. So you end up with parallel lines. Your inhibited line is parallel and above the non-inhibited line. And then the last type of inhibitor that we've seen is allosteric inhibitors. This one we already really talked about already. So you'll recall that in an allosteric or a cooperative binding enzyme, remember we have a weak binding T structure, which has a high Km. And as substrate binds, you gradually get a shift over to a strong binding, lower Km structure, which we call R. And the idea of an allosteric inhibitor is that the allosteric inhibitors make the conformational change from T to R more difficult. Or another way of putting that is they stabilize the T structure. So a well-known example of an enzyme that's allosterically regulated is glycogen phosphorylase. So we're going to talk about glycogen later in the course, but glycogen, it's a complex carbohydrate. It's essentially used inside cells to store glucose. And what glycogen phosphorylase does is it chops glucose off of glycogen. So we have glycogen plus phosphate, that's just HPO4 2-, and that removes a glycogen and gives you a glucose 1-phosphate, which then gets converted to glucose, which can be used as food. So glycogen phosphorylase is allosterically inhibited by ATP. So if we have a graph here of our phosphate concentration, that's our substrate, and the initial velocity of the enzyme, at low substrate concentrations, it goes pretty slow, right? Down here, we're mostly in the T structure, but eventually we switch over to the R. And what you'll notice is that ATP allosterically inhibits this enzyme, by making it so that you have to have more substrate bound for the T to R shift to occur. So that's the structure of the enzyme. It's got 4 active sites in it. And the idea here is that binding ATP makes the enzyme less sensitive to substrate. And this is important. A lot of enzymes that are important in metabolism are regulated by allosteric inhibitors and activators. So what's the logic behind this sort of form of regulation? Well, glucose is a fuel, and what cells are going to do with glucose typically is, in the presence of oxygen, they're going to burn their glucose and form carbon dioxide. And the energy that comes out of this process is used to synthesize ATP, which is an energetic molecule that gets used throughout the cell. So you'll notice that ATP inhibits this process down here. So what's the logic for that? Well, if ATP levels are high, that implies that the cell doesn't need any more glucose. So glycogen phosphorylase has evolved this regulatory mechanism where if there's a lot of ATP lying around inside the cell, it binds to the enzyme and it makes it harder for it to do its job. And that's a way of preventing the cell from wasting glycogen to make glucose when it doesn't need the energy. All right, so we're mostly done with our talk about enzymes, but we are still going to go a little bit further into protein talk later on. We do have to talk about other classes of biomolecules before we go on to functions of cells. So be looking for another lecture soon in which we completely change the subject. And in the meantime, I will see you guys at office hours. Please email if you have questions.